A very good evening to our respected dignitaries, teachers, and dear students. On behalf of the Environment Analysis and Conservation team of Pillai College of Arts, Commerce, and Science, Autonomous, I, Ashwati Gangadharan, cordially welcome you all to our one-day international webinar on current perspectives in environment management. I would request Ms. Deepika Sharma, Vice Principal of Pillai College of Arts, Commerce and Science, to welcome our guests and participants in the webinar. Over to you, ma'am.
Thank you, Aswati. Very warm greetings of the day to all. On behalf of Pillai College of Arts, Commerce and Science, I welcome all the participants and the esteemed speakers, Dr. Nayeem Mullungal and Dr. Seema Mishra, to the international webinar on current perspectives in environment management, organized by Environment Analysis and Conservation team. Pillai College of Arts, Commerce and Science has been accredited with grade A by EnACT in all the 3 accreditation cycles. The college is recipient of Best College Award by University of Mumbai and prestigious ET Now Award for being an outstanding institution, and has received many more accolades and awards. Best recognition of our efforts is the autonomous status conferred by UGC. As an autonomous college, we aspire to bridge the gap between industry and academia, inculcate research culture, and infuse entrepreneurship skills in students. Like, as we all are witnessing very challenging time due to this pandemic, which has disrupted every aspect of life and has shown how sensitive our living environments are. It would be good to follow environment management practices wisely so that we can see a new horizon, giving new ways of living and sustaining in such situations. I appreciate our EnACT team for taking up such a topic which is actually need of the hour. So looking forward to an enlightening session with our distinguished guests. Thank you so much.
Thank you very much, ma'am, for such encouraging words. I would now request Professor Gopkumar Pillai, the convener of the seminar and in charge of the Environment Analysis and Conservation team, to give the opening remarks. Over to you, sir.
A very good evening to all the respected dignitaries and dear participants who are attending this international webinar on current perspectives in environment management. Today's webinar is organized by the Environment Analysis and Conservation team of our college. This team was formed in 2014 with the goal of making an eco-friendly and environmentally consciousness campus. The team comprising of students and faculty members have been putting in constant efforts to promote consciousness, preservation, and protection of the environment. Various activities like canteen waste and garden waste composting as a part of solid waste management projects, energy conservation measures, and environmental awareness programs, training programs, talks, and seminars are being conducted under the roof of EnACT. Today's webinar is on the theme of current perspectives in environment management. As we all know, in the past few decades, the world has woken up to threats of the very survival of humankind due to pollution, environmental degradation, depletion of resources and forest cover, and climate change. Among all these, global warming leading to climate change has become a key concern for the 21st century. With climate change, temperature in the troposphere, over land and in the sea rises, water vapor increases, sea ice, glacier, and permafrost, they lose volume. Oceans maintain heat and energy, and sea levels, they rises, occurs due to the expansion of water and the melting of glaciers. Linked to the interaction of these natural and human factors, extreme weather events like cyclones, droughts, and landslides are more frequent, and they can happen with stronger effects. Climate change will increase the risk of death, injury, and ill health, and disrupt livelihoods and damage property, infrastructure, and settlements in low-lying coastal zones. Climate change will seriously impact global food production as well, as drought, increased unpredictability of rainfalls, and rising temperatures would reduce global crop yields, while warming and acidification of the oceans would affect marine wildlife and fisheries. So immediate attention has to be laid on the problem of climate change if we need to achieve sustainable development. The response to climate change has to be through both adaptation and mitigation. This seminar would provide a platform for the students, academicians, and researchers to engage in an informed debate on the aspect of climate change as a key environmental issue. The discussions and deliberations in the seminar would help the participants understand the potential effects of global climate change and ways to mitigate it. It would be a very informative webinar, and I'm sure that all of you will have an enriching experience through this webinar. Thank you. Over to you, Ashwin.
Thank you, sir. I'm very thankful to our eminent speakers, Dr. Seema Mishra, Dr. Nayeem Mullungal, for taking time from their busy schedule and accepting our invitation. I would request Mr. Gopkumar Pillai to introduce Dr. Seema Mishra. Over to you, sir.
It's my honor and privilege to introduce Dr. Seema Mishra, madam. Dr. Seema Mishra is the director of SIES Indian Institute of Environment Management, Nerul, Navi, Mumbai. Dr. Seema Mishra is a trained professional in the areas of climate change, waste management, biofertilizers, phytoremediation, renewable energy systems, and natural resource management. Her research areas are climate change vulnerability assessment, application of beneficial microorganisms in waste and resource management, low-cost technologies and application in rural areas. She's working with onion and grapes growing farmers of Nashik for improving the product yield, storage quality, and reducing post-harvest losses. She is extensively working in the areas of developing climate resilient microbial products for agriculture. She is consulting agrochemical industries in establishing biofertilizer labs and developing microbial products for crop management. Dr. Mishra is assisting industries in carbon neutrality and renewable energy projects. In academics, she teaches natural resource management, environmental sustainability, environmental compliance, and regulatory affairs at postgraduate level. Dr. Mishra is a certified expert in climate change and environmental sustainability from CII. She has received Konark Memorial Award for Excellence in Research in Biosciences in 2013. Dr. Mishra has also authored and edited books and published a number of research papers in international and national peer-reviewed journals. She was the editor of the International Journal of Environmental Sciences from 2011 to 2016. She is also the member in Board of Studies for Environmental Science at University of Mumbai and an expert member for climate change at State Knowledge Center of Maharashtra in Climate Change, Government of Maharashtra. She is life member of several environmental societies and fellow of leadership in sustainable environment and development. Thank you, Seema Madam, for accepting our invitation and finding time out of your busy schedule to share your knowledge and expertise in this webinar. I now leave this platform to you, Seema Ma'am. All participants are eagerly waiting to listen to your talk.
Thanks, Gopakumar, for inviting me for this international webinar. And it is my privilege to discuss on this platform about the problems related to climate change and management of different resources. So I will start with my presentation. So is it visible? Screen is visible to all of you?
Yes, ma'am.
So as we are progressing, we can see that a lot of issues are there related to climate, and it is expected in 1992 itself, it was expected that the impacts of climate change will be enormous and it will affect all the countries in different continents. So the impacts of climate change we cannot visualize, or it is not very fast change that we can see over a time. But these impacts are very slow and it requires a lot of uh, estimation, evaluation for developing adaptation and mitigation technologies. So today I will cover challenges of climate change and associated problems on mangrove ecosystem and role of rhizospheric microorganisms in mitigation. So when we compare weather and climate, both terms seems to be same, but it is not like that. Weather is a short term phenomena describing atmosphere, ocean and land conditions hourly or daily, and climate is a long term average. So when we talk about climate change, it is not 5 years or 10 years, it is more than 10 years, 20 years phenomena. So when we talk about climate variability, it is generally more than 5 years or up to 10 years. So weather is a short term phenomena of changes in the climatic conditions. So it is not dynamic, it changes periodically, diurnally, even monthly level, annual level. From time to time, we see that the rainfall pattern is changing. We are observing that the rainfall that is required for 1 month is taking place within a day. So when these type of situations are there, it is very challenging to manage our resources, to manage our different type of infrastructure, because the impact of heavy rainfall is enormous on ecosystems as well as on different infrastructures. So climate change is defined by IPCC as the change in climate over a time, whether due to variability or due to any human activity. A change of climate that is attributed directly or indirectly activity that alters the composition of the global atmosphere, in addition to natural climate variability observed over different periods. So basically, climate change is a change in the state of climate, and we can analyze it over different periods, or we can evaluate the impacts of its effect on different type of ecosystems. So climate change is dynamic as well as it is having some impacts. So if we see data of last 40 years, we can see that carbon dioxide is raised. Carbon dioxide basically it is one of the second gas in the atmosphere, and currently it is at second position. But our primitive atmosphere during the origin of Earth, at that time it was number one gas in the atmosphere. So the heat absorption capacity of carbon dioxide is highest. That is why it contributes to maximum heating of the Earth. So when the concentration of carbon dioxide increases, it poses lot of risk to the Earth, and it becomes very difficult for living organisms to survive. So in last 40 years, the level of carbon dioxide has risen. Also, temperature is fluctuated, and it is very difficult. In coming time, it is going to increase further. And fluctuation of temperature is an alarming symptom, because whole the phenology, the life cycle of plants, life cycle of animal, everything is affected. So it is very important to curb this problem at the beginning, and support in the mitigation of climate change effects. So the major causes of climate change are 6 gases that are recognized by Intergovernmental Panel on Climate Change during Earth Summit in 1992. And these gases are carbon dioxide, methane, nitrous oxide, halofluorocarbons, different perfluorocarbons, then different sulfur compounds, they all are related in causing, they have enormous heating potential or global warming potential, and due to that they are contributing lot of heat to the earth. Due to that, temperature is rising. So it is very important to check the emission of these greenhouse gases from different industries or different sectors. So maximum emission of greenhouse gases are from power sector and basically thermal power plants. Then land use changes when we go for deforestation or changes in the landscape or changes in the agriculture activities, or when we go for industrialization, especially where a lot of coolants and steam generating towers, chimneys are installed. Then transport, from transport are substantial emissions. Then building constructions, then other energy related activities and waste management. So all these areas contribute a lot towards greenhouse gas emissions, and it is very important to check the problem and solve this now. So you all might be knowing about the greenhouse effect. So uh, when these greenhouse gases, basically they accumulate near tropopause of our different layers of our atmosphere, and they form a kind of a blanket or shield. And when solar rays pass through it, some of the rays, as you know, that of visible range as well as infrared rays, they reflect back to the atmosphere. So these reflected rays actually helps in maintaining the heat budget of the earth that is required for maintaining different cycles on the earth. But due to these GHG gases, the absorption of heat is increasing, and it is causing heating effect or heat island effect on the earth. So this phenomena is actually, it takes place, it takes us towards climate change. So the effects of climate change, if we can see, it is melting of glaciers. Due to that, sea level rise is taking place. Then uh, precipitation intensity as well as frequency, it is also changing. Due to that, flooding and drought, all these phenomena are taking place. Then natural fire hazards are also very common, not only in America or in other countries, but also in our country. In last 2 years, fire hazards were recorded from Uttarakhand as well as from Jammu and Kashmir. Then agriculture, the impact on agriculture is biggest because it affects the food supply as well as it also increases the prices of all the commodities. Apart from that, different type of biodiversity are also affected due to changes in the climate change, and it is very important to control this at this moment and support in mitigation. So if you see the expected impacts of climate change in India, you can see that average temperature rise up to 2 degrees predicted up to 2100. Then 1 to 4 degrees centigrade extreme ranges will also be there. Of course, these changes will not be same everywhere. It will be variable. As you know that our country is uh, very diverse. It is having arid, semi-arid areas. We have Deccan Plateau. So everywhere topography is not same. So wherever less vegetation is there will be the impacts of temperature will be more. Then increased annual precipitation is expected. So lower frequency of rainy days and increased intensity is expected. Then cyclonic disturbances, the cyclones are regular phenomena on the earth, but now the intensity and frequency of storms will increase, and due to that, lot of disturbance will be there in different ecosystems. Sea level rise up to 1.3 millimeter per year on average is expected. Then fresh water supply will be also affected. Forest and natural ecosystem, because due to high CO2 in the atmosphere, they will get enough opportunity, or their photosynthetic rate will rise, and due to that their biomass production will increase. So their growth will improve, their yield will improve. But due to this excess growth and yield, now their demand for resources like fertilizer or nutrients and water will increase. So a lot of pressure will be there on the natural resources. Then shifting of forest from borders will take place, in terrestrial forest also from boundary movement will take place. In mangrove forest, the movement of forest from seaward side will take place towards landward side. So this phenomena is not good in coastal area because the shock absorbent capacity of mangrove forest will be reduced. Then human health will be impacted. Different vector-borne diseases, different other type of diseases will rise, and it will be having lot of problem in controlling these problems. So then reduced agricultural productivity is also expected. In some crops, it is expected that product will be more, but as I have told you, demand of resources will increase. And our, if you see our agricultural land holding, so maximum farmers are marginal or small. So in that case, it will be very difficult for farmers to generate that much resources that will require for this agriculture. In coastal area, the impact on sea level rise, changes in storm surge and precipitation, fluctuations in sea water and temperature is expected, and ultimately it will affect the mangrove forest and associated biodiversity. So when we go for the climate change mitigation or climate carbon neutrality at the industry level or at the level of the society, the most important aspect is to go for first energy efficiency, to install LED lights and energy efficient appliances, then installation of renewable energy, and third is plantation. So at the level of natural ecosystem, plantation plays very important role, and as a common people, it is our duty to go for plantation, and whatever forest area we have, we should conserve it. So mangrove forest is an interface between land and sea, and these are specialized ecosystems developed along estuarine sea coast and river mouths in tropical and subtropical regions of the world, mainly in the intertidal zone. They serve as a link between terrestrial and marine ecosystem. Ecosystem and associated biodiversity are under the influence of both marine and freshwater conditions. Apart from their diversity, they are also very good in its productivity. Their productivity is very high if they are not having any negative impact like anthropogenic factors or any industrial activity. They are rich in very diversity and flora and fauna. They are source of raw materials for different industries, and due to their rapid mineralization, their sediment is very rich in different nutrients, as well as CO2 sequestration capacity is also very high. That is why mangrove ecosystem play a very important role. Then environment management aspect is also very important because they are buffer against different type of natural hazards as well as erosions. They are source of livelihood. They act as a carbon sink. So they are very important, and it is our duty because in coastal area, it is very important to conserve these mangrove forests so that we can save ourselves and other society from the negative impacts, and because they act as a buffer. So area under mangrove forest, if we see all the dark greens, you can see are showing highest or maximum amount of mangrove forest, and all the light ones are showing the less. So in India, you can see around 7 states and 4 union territories are having mangrove forest, and it is very important to check their health status, their diversity, so that we can go for their proper management. About 40% of the world's mangrove cover is found in Southeast Asia and South Asia. The mangrove cover in India is 4,975 square meter, which is 0.15% of country's total geographical area. An increase in the mangrove cover as compared to 2017 assessment is 54 square kilometer. So in our Thade Velapur Creek also, the density of this mangrove forest has increased, and main reason of increase was presence of nutrients in the sediment. As you know, that different industrial effluents are disposed in sea, and in sediment they provide nutrients to different type of uh, blue-green algae, phytoplanktons, as well as to these plants. But in long run, when sedimentation will increase, it will be posing lot of challenges to the spread of mangrove forest. So sedimentation rate, if it is high, it will also not good, because it will affect the mangrove forest. So expected threats on mangrove forest due to climate change are sea level rise and impacts. So sea level is expected to rise from 0.28 meter to 0.98 meter by 2100. And you can see that all the dark areas in this map are showing that sea level rise will be very high in the areas. In India, the impact of sea level rise will be very slow, uh, but it will be visible in West Bengal, Odisha, and Gujarat areas. In Maharashtra, its impact is less than West Bengal. So, increase in flooding incidences will lead to the plant death in seaward margins, shift in species composition. Trees in microtidal areas will be more vulnerable. The sedimentation and its accretion will be more because of slow velocity of water through aerial roots, litter fall, and deposition of filamentous algae. So the measurement of sediment accretion is essential for determining changes related to sea level rise. So it is very important to check the sedimentation rate year, so that it will be very clear if sea level rise is happening or not. The severity and frequency of storms will be more, and its impact will be more on the seaward side of the mangrove forest. So uprooting of trees will take place, and trees will become very fragile, and due to that there will be lot of risk. Because in Mumbai, if you see, in last 40 years, out of 15 species of mangrove, only 2 are dominant. And this is not good, because when you go in any forest, the positioning of different trees actually depends on their capacity to absorb wind effect or other natural calamities. So some species prefer to be on the border, some species will be inside. So when any forest is having only single species, then its capacity to face the pressure will be less. So it is very important to have a multi-species forest, and in our city, this is a very big challenge. Then variability in precipitation we are observing this year also. In June, very less rains were there. In July, there was rain. In August, September, it's also rained. But in September, in one day, we have got rains of full month. So these type of variations are not good, because when water more than the capacity of any ecosystem goes into that, it affects the natural living organisms. So plants or animals, they all will be affected. Salinity will be changed. So animals will have very difficult to cope in that changing scenarios. So alteration in seasonal average salinity will be more in the fringes near estuary and in the interior of the mangroves. And decrease in the precipitation is equally very harmful, because it increases in evaporation and needs to increase in the soil salinity. So excess salinity will also be not good, and it will affect the seedling survival. So most of the natural forests are self-propagating. So when there will be excess of salts or excess of any other heavy metal, it will affect the propagation or self-propagation of these species, and it will be really very challenging. Then variability in temperature is also expected, and it is expected that it will increase by 4.8 degrees centigrade. So mangrove species composition, phenology, productivity, ultimately the latitudinal ranges and distribution will be affected, and assimilation of carbon dioxide will also be affected. So with rising temperature, it is expected that photosynthetic rate will rise, but due to that, at some places it will decrease also, but it will ultimately it will affect the sediment quality as well as availability of other resources. So apart from climate change, the other stressors on mangrove forest are natural factors like invasive alien species. Invasive alien species of plants and animals both pose a risk on the ecosystem in maintaining its dynamics. Then anthropogenic activity like reclamation of SEZ and infrastructure project, domestic sewage discharge, industrial activities like industrial effluent discharge, solid waste dumping. So the ultimate impact on mangrove forest will be its depletion of nutrients, decreased productivity, reduced mangrove forest cover, adverse effect on flora and fauna. So as I have told you, natural causes are having enormous impact on the mangrove ecosystem. So with this objective, we have conducted comparative assessment of native as well as invasive tree species in mangrove forest of Thane Belapur Creek and Thane Creek. As you know that maximum density of mangrove tree Avicennia marina is there in Thane Belapur Creek or in Thane Creek, and we have taken one exotic species or invasive species, although there were many. The main agenda of comparison was under, as you know, that under Aichi biodiversity targets of Convention on Biodiversity, as well as in UN Sustainable Development Goal 15, focus is on invasive species. So it is very important to find out how invasive species are posing challenges to our present native trees species in these mangrove forests. So basically, we have selected two distances, 50 within 50 meters from the sea level and up to 300 meters from the seaward side. So we have observed that mangrove species, which was within the distance of 50 meters, was having very good DBH, height, and other parameters. And the mangroves of Belapur Creek were not having that much good quality in comparison to Thane Creek. And if we compare with invasive tree species, although there are many, like Lucina leucocephala, Prosopis juliflora, we have taken only Lucina leucocephala, and we have observed that the height, girth, everything of invasive tree species was more than the mangrove trees. And due to that, they were causing shadow effect on the mangrove species. And we have observed that there was a lot of changes in the dynamics of their growth, productivity, as well as rhizospheric microorganisms. We have observed that the carbon dioxide sequestration capacity of invasive species was more in Thane Belapur Creek in comparison in Thane Creek in comparison to Thane Belapur Creek. And if we compare it with mangrove species CO2 sequestration capacity was not very high, although they are known to have a very good capacity, but due to the changing dynamics or competition between the native species and invasive species, mangrove species were not able to perform. There may be different reasons. Some of the reasons, important reasons, are shadow effect, and due to that, photosynthetic rate decreases, and plants are not able to produce that much biomass that is required. You can see here from regression graph that with the increasing distance from the creek in mangrove species, CO2 sequestration decreased. However, in invasive species, it increased. So invasive species are basically having a very good capacity to face any type of challenges or any type of problems or negative impacts. So Lucina leucocephala was introduced in our country during 1980s as a multi-purpose tree species to support the requirement of fuel wood and timber. But now it is increasing very fast in different parts, different ecosystems of our country. And in mangrove tree species, in mangrove forest also, they are growing in variable pH. They are available at pH 9 also, they are available at pH 5 also. So they have a very broad range. So invasive species, as we know, they are very broad in their survival instincts, and that is the big challenge. And it is very important to identify these challenges now and go for its management. So it is very important to first find out what type of reclamation and degradation activities are going on, and we should also try to control it. Then reducing disposal of sewage, industrial effluent, then construction and demolition waste, maintaining tree diversity in mangrove forests, selective felling of invasive tree species. By characterizing rhizospheric microorganisms also, we can promote better or beneficial one, and we can improve the stand quality, basically forest quality, because they provide nutrients, they maintain the biogeochemical cycles. So it is very important to maintain the microorganism health in the rhizosphere. So microorganisms play a very important role, and in mangrove forest also, bacteria and fungi, they constitute around 91% of the total uh, organisms, microorganisms, and algae and protozoa represent only 7 and 2%. Among different groups of microorganisms, bacteria believe nitrogen fixers, phosphorus solubilizers, cellulose decomposers, nitrifiers and denitrifiers, sulfur oxidizers, methanogens, iron reducers, they all are present in mangrove ecosystem. And despite of global advancement, still very little is known about their function and their different type of activities. So it is very important to characterize these microorganisms properly with the advanced biotechnological tools like metagenomics, pyrosequencing, so that we can identify their functional characteristics and we can use them for the improvement of mangrove forest qualities. Another study I want to discuss here is about mycorrhizal fungi. That is also very important phosphorus mobilizer in soil, and it is a very good microorganisms that is present in symbiotic association with plants as well as other beneficial microorganisms. So in this study, in some part, Gopa was also involved. So I'm very happy to share this study here. So actually, we have conducted this study in different degraded or polluted sites of mangrove forest in Mumbai, Navi Mumbai, as well as in Alibag district. So we have observed that in Thane Creek slum relocation domestic site, Visarjan Ghat was selected. From Alibag, fertilizer plant effluent was actually flowing through that mangrove forest we have selected. In Mansarovar, we have selected brick industry discharge. In Mahul, we have selected the area where three refineries, petroleum refineries are there, as well as salt pans are there. Then Aroli Creek, we have selected STP effluent disposal site. In Uran, we have selected reclamation of SEZs and major port. Then in Gorai, we have selected industrial effluents. At Palm Beach, we have selected where C&D waste were disposed. And Bicroli or Godrej mangrove, we have selected as a control for the study. So these are the different pictures of the study. So arbuscular mycorrhizal fungi is symbiotic association between plant roots and very important association, wherein fungi provides different phosphorus. It mobilizes phosphorus. Apart from that, copper, iron, zinc, manganese. These micronutrients are also easily provided to the plants. Plants in turn provide organic content as well as they also release different type of hormones and enzymes, also different alkaloids, and due to that the uh, defense mechanism of plant increases. So this is a very important tool for not only improving the growth and survival of plants under adverse conditions, but also to find out if that area is affected by some type of degradation or heavy metal contamination. So AM fungi is a kind of a bioindicator. So in this study, you can see here they have tripartite relationship between plant, fungus, and soil. So we have observed that root infection, AM fungi infection, we can study in the root of a plant, as well as in soil we can get different spores. So we have observed that the root infection as well as spore density was maximum in Mahul area. So Mahul area, as I have told you, it is petro refinery industries site, as well as that the national fertilizer plant is also there. Apart from that, there are several salt pens. Then we have also observed that at different sites, the root infection and spore density was more in comparison to the control, that was Godrej Vikhroli site. So you can see here AMF root infection in mangrove tree. So we can see vesicles in hyphae of the roots, or we can see arbuscules, and they are very important part for maintaining the supply of different nutrients under adverse condition to the plant. So as you know, roots are very dynamic in death and decay. So when any root dies or decays, these vesicles convert into spores and it remains into the soil till it gets water or adverse conditions so that it can germinate. So different type of AM fungi species we have observed from different type of forest. So these are the spores that we have got from the soil, and these are the different dynamics of this. This is diversity index. So Shannon Linear index was also more in Mansarovar area. That was a brick kiln industry site, and Markov index of species richness was at Palm Beach site, and Paleo species index was also at Mansarovar site. Heavy metal content in mangrove tree sediment, if we see, so Mansarovar was having highest nickel content, followed by lead content in Mahul, and chromium content was also higher in Oran area. So these heavy metals were more, and we have observed that AM fungi diversity was also more in these areas. So this study proves that AM fungi can act as a bioindicator, and they can also use for the reclamation of forest because they have multiple benefits. Then glomalin content. Glomalin is an glycoprotein, and we know that glomalin is secreted by the AM fungi. And wherever glomalin is more, it means that organic carbon content is also more, means CO2 sequestration capacity of soil is more in that area, and it is due to the AM fungi. Apart from that, these glycoproteins are able to absorb different type of heavy metals which are present in the sediment. So nickel and chromium, all these metals can be present binded with these glomalin. So this data also proves that presence of AM fungi more density with glomalin and heavy metal. It shows that mangrove forest, wherever they are degraded, AM fungi diversity is also good. So in future, actually, it is very important to assess the vulnerability of mangrove forest because, as I have told you, there are different changes, geomorphological changes, spatial changes, topographical changes in forest quality. So it is very important to identify what are the different factors that require proper adaptation and mitigation strategy against climate change. It is very important to evaluate microflora of these degraded and pristine mangrove rhizosphere through classical and molecular tools for evaluation of their potential, metagenomic studies to explore best isolates for their multiple applications, and application of best isolates in afforestation programs. So I think this strategy will help in improving the forest trend quality as well as plant growth under adverse conditions. So, so I have completed my presentation, and I hope I have completed on time. 45 minutes were assigned to me, so I think I have completed on time. And if you have any questions, so you can ask me.
Thank you, ma'am, for such a wonderful and informative talk. Before we move on to the next technical session, I would request Ms. Bhakti Hirani to put forward the questions and queries by the participants to Dr. Seema Mishra. Over to
Hello,
you,
good
Bhakti.
evening, ma'am.
Good evening.
So there are a few questions by the students. Like the first one is, can we study mangroves in laboratory, or is it possible to grow them in laboratory?
Actually, in laboratory, a lot of challenges are there because you know that these forests requires diurnal flow of water. As you know, tides are there, diurnally they flow, low tide, high tides are there. And due to that, salinity gradients changes and different nutrients also changes. So it is very important to provide these type of diurnal changes into the forest, then they can survive. Under nursery condition, Godrej mangrove as well as mangrove center of our forest department of government of Maharashtra, they are they are doing nursery raising. But if you want to grow it as a full mangrove forest, then that tidal impacts are required. And in Japan, actually, some scientists have tried, and for that, mechanical systems are required, and that is very costly.
Okay, so we have second question, that is, what can we do to have sustainable species of mangroves in our laboratory or in our environment? How can we make them sustain the mangroves?
Yeah, for making them sustain, it is very important that they should be grown in forest area that is having multiple species, and not only true mangrove but different other mangroves which are herbs or shrubs. They should also grow along with them, because if you go in mangrove forest, you will find Salvadora persica is present around the periphery of mangrove forest, that is having light green leaves. So they are known to actually protect these true mangrove species. So it is very important that multiple species diversity should be there. Apart from that, there should not be any exploitation. Like if we are disposing any waste or any type of activity like cutting is going on or grazing is going on, then it will impact the quality of the forest. Then it will not be very strong to sustain under adverse conditions of uh, very high heavy winds or storms, or due to that other activities. So it is very important to maintain the quality of these forests. Apart from that, rhizospheric microflora, basically beneficial of different types, should be studied, and artificial consortia can be provided to it so that these plants will grow sustainably.
There's last question, ma'am. How can we study the effect of climate change on biodiversity in the forest?
Yeah, actually, this study will not be of short duration, it will be of very long duration. And apart from that, first we have to take secondary data from previous study. On that basis, we should establish our study. It is very important to collect meteorological data of last 20 years to find out what type of fluctuations are there in weather condition. Then it is very important to find out what type of species are there in that forest, what are their economical utility, and also if some forest data is there about their height or girth or nutrient level in the soil, so we can compare it with our study. So in that way, we can identify if temperature is rising, so it is impacting the forest or not, or with the changes in the climatic condition, what is the soil profiling within 10 or 15 years. So this is a very long study and it can be done in this way.
One more question is there, ma'am. Like, if we cut the mangroves from their site of origin and then we try to plant it at different place, it basically dies out. So, what can we do to prevent that from happening?
Yeah, actually, as you know that sediment quality varies from place to place, and if you compare the sediment qualities, you will find lot of pollutants are released into this ecosystem, and as per that sediment quality is there. So when we shift mangrove forest from one area to another area, they take lot of time. Even terrestrial plants also, they take lot of time, and special mechanism is there for their extraction and then their replantation. So these type of activities are required. Apart from that, uh, there should be proper flow of that tidal water. If it is not there, then plant will not survive because uh, under artificial conditions, this plant will not survive efficiently.
Thank you, ma'am. That's it from the
Just a minute.
questionnaires. Over to you, Ashwati.
Hello. Gopakumar here. Yeah, ma'am, I had one question to ask you. Like, how do we measure the carbon stock in mangrove wetlands? Like, how can we access it?
Carbon stock can be measured in two manners. First, I'm just talking about crude manners. Uh, Other methods are also there, but for that, first you need to do profiling of sediment. How much organic content is there, up to what depth organic content is same or it is changing. Then we have to do profiling of carbon dioxide in trees. As you know that we cannot cut these trees, huh? So there is method of allometric equations. So we have to take height and girth of that plant, and we can put that data in allometric equations and find out how much total volume of that wood is there, and it can be converted into carbon dioxide. It is a very simple phenomenon.
Okay, thank you, ma'am. One more question. Like, which are the endangered species of mangroves now? Like, which are towards the verge of extinction?
Yeah, actually, in Mumbai earlier, Rhizophora mucronata was there, Stereops was there. Now they are moving towards fringes, and Rhizophora you will find only now towards Shahar near the estuary of Ulhas River. So they are changing their pattern. So in Mumbai, you will find only Avicennia marina at some places. Avicennia alba is there. Then government is going for afforestation of Sonarshia patella. So that is actually there in Mithi River side. Apart from that, if you go in Aroli, as we have gone, Gopa, so there you will find 4 5 species. But all these species are not dominant. Only Avicennia marina is dominant. So it is very important to grow these species under artificial conditions or in nursery condition, and then go for replantation to prevent their extinction.
Okay, thank you so much, ma'am. It was great to work with you on mangrove project.
Thank you.
Thank you very much, ma'am. And I'm sure that your immense experience and knowledge will benefit the participants in many ways. I would like to invite Professor Bindu Rajguru, member of Environment Analysis Conservation Team, to introduce our next speaker, Dr. Nayeem Mullungal. Over to you, ma'am.
Thank you, Ashwati. Good evening, everyone. I take this opportunity to introduce Dr. Nayeem Mullungal, the second speaker for today's session. Dr. Nayeem Mullungal is a faculty member of Marine Chemistry at the Department of Biological and Environmental Sciences, Qatar University. He holds a doctoral degree in chemical oceanography from the Department of Chemistry, University of Otago, New Zealand in 2017 with an international student scholarship. He has completed his MPhil in marine chemistry with first rank from the Department of Chemical Oceanography, QSAT, in 2011 after finishing MSc in hydrochemistry from the same department. The major research focus for Dr. Naeem is on the oceanic nitrogen cycle and the use of novel stable isotopic techniques. Other research interests are on mangrove biodiversity, marine nutrient cycling, and ocean acidification. He has presented his research works in more than 20 international conferences in 8 different countries. Dr. Naeem has secured various accolades and international awards for his work from international agencies such as AGU, SOLAS, IAGS, NZMSS, and Chinese Academy of Sciences. Welcome, sir, and we are really honored to have you as a resource person today. I now request Dr. Naeem Mullungal to start with the session. Over to you, sir.
Okay, I'm sorry, you couldn't hear me, right? Before? Is it clear?
Yes, we can hear you now, sir.
All right. Just one moment. So thank you so much for the nice introduction. I'm really honored to be here to talk to you people here today, especially on environment. Actually, we start a little bit early, right? So I will talk about the nitrous oxide in the ocean, which is my research topic, and its role in the global climate change a little bit on that. And today here I'm gonna focus on the Arabian Sea work that I have done around 5-6 years back. So here we go. I would like to take you for a voyage actually, and we'll be back to the lab, and then we'll discuss about our results. So here, my talk is like this. We go through the importance of nitrous oxide in a global perspective. Then we go to the importance of nitrous oxide in deep in the ocean, and I will talk about my sampling, my analytical processes, and we'll discuss about the results. That's what my plans for today. So, though we say nitrous oxide as a laughing gas, it's not a matter of laughing actually. The major importance that I'm not going to focus on other greenhouse gases, but at the end of the talk, I will explain a little bit. I will focus on the other greenhouse gases and how their study will be a little different from what we do with the nitrous oxide. But I hope already Dr. Mishra very well explained about the mitigation strategies and technologies in general on the global warming and climate aspect. So I don't want to go through it in detail again. So as we know, like the first thing, the nitrous oxide is a greenhouse gas. But why it is important than carbon dioxide? It's not important as compared to carbon dioxide, but what makes it important? It has a 300 times global warming potential than carbon dioxide on molecular basis. That means it can trap more heat than 1 molecule of carbon dioxide. Second thing, its lifetime, average lifetime is around 200 years. So it stays in the atmosphere for a long time. And the second important thing, this is a precursor of ozone depletion in the stratosphere. As you can see in the right figure, the figure in the right side, you can see how much it contributes to the global warming. So ozone depletion, after the Montreal Protocol, we know we don't use much chlorofluorocarbons. So then the second one, or nowadays the most important one, which caused the depletion of ozone, is nitrous oxide. And this work actually got Nobel Prize in 1990s. So now if we look at the nitrous oxide concentration in the not only nitrous oxide, all greenhouse gas concentration in the troposphere, you can see it here. You can see the carbon dioxide concentration, the nitrous oxide concentration, the methane concentration, and the chlorofluorocarbons as well. This all in the last 40 years, you can see an incredible increase by time in each year. But it was not the case before. Like if you consider this from last 1,000 years, you will see that this increase is from last 250 years or last 200 years. That means after the Industrial Revolution. And that's very important. That's the importance of nitrous oxide study in even 2020. And in future, it's already proposed that there will be an increase of oceanic nitrous oxide because there are increasing suboxic and anoxic coastal ecosystems. Oxygen minimum zones are increasing. The eutrophication is supposed to be increased in the coming years through the sedimentary denitrification. Plus, the deposition of aerosol and nitrogen and species, the increase in productivity is also suggested. Plus, to add this, there is a paucity of data for N2O as compared to the other greenhouse gases. And so I just put two, screenshots of two important reports. See, look at this. Ocean source of greenhouse gas has been underestimated in terms of nitrous oxide. This is a recent study from MIT, and you can see the other one. This is a 2014 research paper as well. Nitrous oxide emissions could double by 2050. So that's really alarming. So that to point towards the importance of the work in N2O. So from where this N2O is coming to the atmosphere. If you look at this, you can see around 30% is coming from the ocean environment, including open ocean or marine environment, 30%. And you can see the most important other natural source is soils. And soil and open oceans or the oceans contribute the natural source, while you can see the biomass, biofuel burning, agriculture, industrial activities, and agriculture contribute to the rest of the percentage. But then, see, this is actually the one I have also deduced from my PhD thesis. Actually, you can see it, the relative contribution. You can see soils, oceans, then all the other colors you can see in the chart, they are from anthropogenic activities mainly. So why it is important for us? Because normally nitrous oxide is released into the nature to the environment by breaking down the nitrogen in the soil and oceans. And as you can see, the natural source is double that of the anthropogenic. While, even after the industrial revolution, the natural emissions not changed significantly. But then what have changed is the man-made emissions. So this increase caused the increase in the atmosphere to rise steadily by last century. So now, again, I will go. So now we know the importance, the source, and the scenario. And now how I will go into the mechanism in more detail, how it is formed. It is especially when I'm going to look into the oceanic perspective now. I'm not going to talk about the soil terrestrial environment only or human anthropogenic activities. I'm going to focus on the natural production from the ocean. But if natural production from the ocean is increasing, that means there is something else happening that we will talk at the end of the talk. So the conversion of nitrogen to bioavailable nitrogen forms and the subsequent transformation between these different species are the central to the nitrogen cycle in the ocean, as you know. And this is this is driven by the organisms that reside in the ocean. So what are the major processes? Earlier, it was suggested only nitrification and denitrification, but later, in last 10 years, the research also point towards the archaeal production. So you can see it here in this PowerPoint. You can see the nitrification in the left-hand side and the process of denitrification. And there is a coupling between these two processes. Sometimes it is also proposed that this nitrifier denitrification or this coupling process is also a precursor of nitrogen and nitrous oxide. And also have a look at this process. This we will focus on this figure later when we talk about the pathways responsible for the production. So you can see ammonia, the conversion of ammonia into nitrous oxide during its transformation into nitrate in 3 different ways. So I have named them as pathway 1, pathway 2 and pathway 3. You can see when it is formed from NH2OH, that's the pathway 1. But if it is formed from the nitric oxide, NO, then I call it as pathway 2. And pathway 3 is the formation from nitrite we want to differentiate between which method is responsible, which process is responsible for the production at the end. So as you can see, I don't want to go in detail about this. You all might be aware about what is nitrification process and what is denitrification process. But you can see from here, during the nitrification, which is the bacterial oxidation of ammonium, you can see that this got some link with the oxygen, an inverse relationship with the oxygen, and that's all about the process. Now the estimates from the general, the estimates based on the existing studies, you can see here that open oceans contribute 40%, while the coastal upwelling zones and estuaries or continental shelves, look at these, they occupy only a little bit portion of the ocean as compared to the open ocean, but they contribute comparatively larger than their area. So what might be the reason? We can answer it at the end. So, you know, what are the factors? Any factors that will affect the nitrification or denitrification will have an effect on the production. So it is mainly, as I told you, the concentration of oxygen is one of the important factors. There are nutrients, there are physical factors like wind, mixing, etc. But the most important factors are nitrification and denitrification. So before we go ahead, I just want to quickly go through the arguments and questions existing in this study. There are a lot of debates going on, like, you know, there are climate change deniers. Same way there are debates in the nitrous oxide or ocean chemistry. A group still is of the opinion that it's mainly produced through nitrification, and some suggest it's mainly through denitrification. But we will look into our results at the end to see what is the major reason. And my study focus on the Arabian Sea actually, though I have done in Pacific and different regions of the Pacific too. So the main focus for us today is our study area, a source or a sink of nitrous oxide. And the next question, how it is distributed in this water column, how is the distribution along the oceanic water column, and how it varies with the dissolved oxygen. And I will answer to the question, which is a debate, which process is responsible for the production. So why Arabian Sea is important? You know, the Arabian Sea is the most intensely studied region with respect to nitrous oxide among the world ocean. There are some other regions in Pacific Ocean as well, also in Atlantic. Similar way, Arabian Sea is one of the intensely studied region. Another thing, the Arabian Sea is only 0.43% of the world ocean, you know, it is contributing around 20 to 35% of the N2O global flex. So what might be the reason? The major proposed reasons are the, you know, existing extremities in the wind stress and, you know, different upwelling and upwelling conditions. That's a special scenario which is present in Arabian Sea in particular. This can be a reason. Plus, we'll see how the coastal values that might be that might be a source from human activities through the, you know, nitrogen inputs from the fertilizers which we use on the land. But why my study is unique from the other Arabian Sea studies? There are very few studies on the stable isotopes in the Arabian Sea. Stable isotopes are like, you know, novel traces these days for study any kind of mechanism or process studies. Plus, there is another thing called isotopomer. I won't talk a lot in detail about that. I will just give you a clue about how my results, and as it's not published yet, these results, and it's just underway. So how the third one is the isotopomeric data, which is you know, not at all done in detail in any part of the Arabian Sea until so far. So that's our focus of the study. Most of the studies until now in Arabian Sea measured and estimated concentrations, and they also talk about the flux from ocean to air, and they also focused on dissolved oxygen and nutrient modeling. But there is a lack of novel techniques which is used in other study regions of the world, like off Chile, of Mauritius, or Aloha stations of United States. So and we have a lack of proper studies based on the biological methodology as well here. So first of all, before I go ahead, I will give you a little a basic clue about the stable isotopes in the how they are used in the ocean science. There are mainly two stable isotopes of nitrogen. So we have N2O, NNO. That's what the structure of N2. I will show it later too. So there are two elements, nitrogen and oxygen. So there will be for sure two different stable isotopes of nitrogen and oxygen. And for nitrogen, there are two stable isotopes, 15 and 14. And you know, the major concept here of stable isotope, we know there are radioisotopes. We don't talk about radioisotopes. We are talking about the stable isotope, the natural abundance ratio. There will be a natural abundance ratio, and that ratio 1/274/2, which will be almost constant that's called a delta N15. That will be almost constant in the nature. So if it is not, if there is any changes in this, that's a big clue about some existing features in that region. Similarly, oxygen got 3, and these 3 are 16, O16, O17, and O18. So just to give you some basic idea. Maybe some of you are not much interested. We measure this using a special equipment called, it's a mass spectrometer, isotope ratio mass spectrometer. And this is expressed as you can see here in the figure, delta N15, which is for nitrogen we use in terms with respect to air, while for oxygen we use Vienna standard mean ocean water reference. So in general, the isotopic composition of nitrous oxide is helpful in understanding the important sources that are isotopically distinct. That means you have nitrous oxide in the air that goes to the water. When it goes to the water, it may undergo nitrification, it may undergo denitrification. If sometimes there may be no process at all. So depending upon all this, the isotopic signatures will change, and that is the best clue we can have about its source actually. Because it will help us to know what is the process that add N2O or remove N2O from the ocean. So the delta N15 of N2O in the air is 6.72 plus or minus 0.12 per mil, and that for oxygen in the N2O is 44.62, as you can see here in the slide. So what is that? That's the mean value in the air. When the N2O enter from the air to the water, oceanic water, if there is no other biological process, same N2O enters to the water, there is no formation or dispersion in the water, then that means it will not have big change. So if you take a surface water, took the N2O isotope measurement for N2O, if it is close to 6.72 and 44.62 within this range, that means there is no other process. It's the N2O which came from the ocean. But if it is decreased or increased, then there is something. It may be because of nitrification, or it may be because of denitrification. See the chemical reactions and the physical processes. It's called like isotopic, we can talk about it later. So chemical, at the end of the talk, I will go to this now. So the chemical reactions and the physical processes mainly controls the isotopic composition of nitrous oxide during the biogeochemical cycles. How? See here, the light isotopes, we know N15 and N14, where the light one is N14 in terms of N, and O16 is light in terms of oxygen. They are preferentially taken up over the heavier isotopes by microbes, and it will be transferred to the reaction products. That means when you have a product during the nitrification or denitrification as N2O, N2O can be a byproduct or an intermediate product during this nitrification or denitrification. So when the N2O is formed from this, the nitrogen that is present in the N2O will have isotopically lighter heavier ones, because the other ones will be taken by the microbes. So this is because during the microbial process, the molecules with heavier isotopes will react very slowly. So as a result, what will happen? The substrates that will be become enriched in the heavy isotopes, and the products will be depleted with respect to the. So sometimes I use the word dual isotopes. That refers to the N15 and O18 of the N2O. When I say dual isotopes of N2O, that means N15 and O18 together. So as I told you, N2O is both a product of nitrification and denitrification. But same time, during denitrification, which I didn't mention at the beginning, during denitrification, N2O is also consumed. That's the only exception between denitrification and nitrification. Nitrification only causes production of N2O, while denitrification causes consumption as well. So N2O, we know it's a product of nitrification or denitrification. Now we know this very well. So what is nitrification? Formation of nitrate. Denitrification, conversion back to nitrogen. During this process, what's happening? We know the substrates are becoming enriched in N15. So the products will be depleted. So if the N2O is produced through nitrification or denitrification, what will happen? That N2O will have less signatures of isotope. We say also isotopic signatures for the isotope value. So the N15 and O18 of N2O produced from nitrification or denitrification will have less value than the standard air value. That's the first clue, which is very important actually. But the problem, denitrification also results in consumption of nitrous oxide. That will be if as a result of denitrification, N2O is utilized, then the reverse will take place. As a result, if you see N15 or 18 signatures of N2O like 15 or 20, that means there is denitrification. And so we can say that the dual isotopic signatures will give you a good clue about the process. Now, but they will not give you any information about this one I told you, this pathway. This I'm gonna show it again, which is like from where this is coming. If it is through nitrification, is it from pathway 1, pathway 2, or pathway 3? This is nitrification. In this nitrification itself, N2O is formed through 3 different pathways. So to get a clue on this, the dual isotopic signatures will not help you. So what you will do for this, we can use the so we can use the oxygen isotopes. So for how we use oxygen isotopes for this study, I'll show you. For this, you take dissolved oxygen in the water, you use the simply water oxygen isotopes, and you will use the dissolved oxygen isotopes, and then you will use the nitrous oxide oxygen isotopes. So how it is that, how it is done, see here, look at this figure. When ammonia converted to hydroxylamine, you can see that oxygen is coming from dissolved oxygen. While when NO converted to NO2, there is an addition of oxygen from water itself, not from the dissolved oxygen. You have to remember this. When we say seawater, dissolved oxygen is different from the oxygen in the H2O. Okay, so this is a very important clue. So if this N2O is formed from NH to OH, the O in N2O is simply coming from dissolved oxygen. But if I have the second pathway N2O, it is again same. But if I have N2O from the third pathway, then that's different. See, it will be an oxygen coming from a combination of both dissolved oxygen and H2O delta O18 values. So we need to know that delta O18 value or O18 oxygen isotope value for dissolved oxygen is different. That for water oxygen is different. That for nitrous oxide oxygen is different. When we know these three different values, just very difficult to actually to do all this analysis after sampling. But if you know this, this is a really excellent data to comment on this. And then the next one I did was isotopomer studies. What is isotopomers? It's like, there are a set of molecules having isotopically substituted atoms. They are same molecules, but their isotopes will be different. For example, look at this N2O. This is the structure of N2O. You can see it here. And this I also want to tell you, the nitrogen which is very close to oxygen, it is called alpha nitrogen, and the nitrogen which is the second nitrogen which is far from the oxygen is called the beta and beta nitrogen. So actually N2O can have 12 isotopomers, but only significant ones are 5, and they are here. You can see the oxygen. These are the ones, you can see it. What is the difference? The difference is only in their isotopes in their molecules of isotopes of nitrogen and oxygen. So N2O, as I have shown you, is a linear molecule, and the analysis of intramolecular, that's called the analysis of intramolecular distribution of N15 in N2O. That's what isotopomers. You wanna know what is the value of alpha nitrogen and what is the value of beta nitrogen. That will be different. And this will give you a very important clue about the process as well, because this is independent of the processes. So that will, for denitrification, there will be a specific isotopomer value. For nitrification, there will be another specific isotopomer value. So how do I find this intramolecular distribution? This is called site preference. And what is site preference? If I reduce the delta N15 value of alpha nitrogen or delta N alpha from delta N beta, that is what the site preference is. So the measurement of site preference of nitrous oxide will allow you to identify the mechanism of N2O formation by different microbial pathways, as I have told you earlier, because it's independent of the nitrogen isotope ratio in the starting material. But in other case, when we talk about dual isotopes, N15 and O18, that is source dependent, substrate dependent, while this one is independent of the starting material. It has a unique characteristic depending upon the biological process. So these are the major things before we go ahead with our voyage. So it was like a pre-voyage briefing. Now I started my trip here actually. I started from Kochi, and these are the 3 stations I did collect my sample, and you can see the geographical location of the 3 stations, and they represented upwelling during the time of sampling. And you can see myself collecting the sample. I used the Tygon tubing for use of, I took actually the first sample. If we are doing sampling, this is called CTD rosette. If you're not from, if any of you have no clue about how marine sampling is done, this is a special bottle which will be sent to the bottom of the ocean, and then it will collect samples from respective depths. And once that is back to the deck, we know from where depth each of the bottle coming from. And then I, carefully, without any air contamination, I use special serum vials here, you can see, and I have special crimpers to seal them. Then, once the samples are ready, I'm back to the lab. I collect a lot of samples. I need samples for concentration. I need samples for N15 O18 measurement. I need samples for oxygen isotope measurements of water, of dissolved oxygen, and already I will get to O18 for N2O from the dual isotopes. So this is the actual setup we need for the analysis. And my first year of PhD was production of this equipment, and I did that, and we were successful in recreating that, which actually this is a 2010 method. I started my PhD in 2011, and we were successful in completion of this equipment production with the help of our workshop in 2012. And then we got heaps of data from this one. So that's I just wanted to show you how I did the measurement. So you can see the one at the bottom here, that's the isotope ratio mass spectrometer, meter IRMS. The ones at the top here all are created by ourselves. So how was the distribution of N2O in the water in the Arabian Sea from these three stations? You can see this. Can you see my pointer when I'm using the pointer? I don't know, like. Anyhow, so the water column distribution showed a single peak structure. You can see a single peak. But in other regions, Northern Arabian Sea, if you do the same study, you will see a double peak structure, because in Northern Arabian Sea, there is a very huge denitrification zone. So when you have a denitrification zone, what will happen? At that region, there will be huge consumption too. So as a result, N2O concentration will decrease. That's why there is a double peak structure. Here, there is no denitrification zone. So when there is an oxygen minimum, there you will observe more nitrous oxide. And this is with respect to the earlier studies, our values were really in good agreement. And then you can see the upwelling stations had very high concentrations. The UC, the Cape Cameron sample was, the Kanyakumari sample was showing very high concentration, while the one of Cochin SK, of Cochin SC was little lower. But all of these stations showed very high surface concentration than expected, or than the air values. But in the upwelling regions, you can see the green ones are non-upwelling station, while the other two are upwelling stations. You can see the difference between upwelling stations and non-upwelling stations. So you can see here the dissolved oxygen concentration. And when you look at the PowerPoint, you can see that if you compare this, have a look here, and then have a look at here. These two have an inverse relationship, which is according to, it's a preliminary proof that the process responsible for N2O production is nitrification, because during nitrification, there will be oxygen utilization, and it's more oxygen will be utilized as nitrification increases along the water column. So oxygen is decreasing, same time N2O is increasing. So and also the maximum concentration of N2O was observed where the minimum of oxygen was present. So that's a very good clue about the production of N2O through nitrification. It's not like a valid proof. Earlier days in 1980s, they considered this as the important clue, but later it is considered as only a basic clue. You can say, because there are exemptions too in other part of the world. So throughout the water column, when you look at the surface, emission, if you want to see whether this region is a source or sink, now look at the emission. You can see it is super saturated. N2O saturation, you can see all over the water. It is more than 100%. Even non-upwelled stations show a 150 saturation. But upwelling stations showing a saturation of 200 to 300. So surface to bottom, this region is a very significant source of N2O, but it's only based on saturation. So now we can do emission calculations. Might be you are aware about these calculations, flux calculations from the previous talk. I'm not very sure. We use some equations, physical oceanographers equations for air flux calculations, C2 air flux calculations. I employed 3 different equations from different studies. From all the equations, you can see that it is a very huge concentration in teragrams of N2O per year as compared to the many other part of the oceans. So initial feedback for us from the process, to get a more good picture of this, I calculated a parameter called delta N2O and apparent oxygen utilization. Apparent oxygen utilization is calculated using dissolved oxygen values. Delta N2O is calculated by using the surface concentration and the equilibrium concentration. And from this, you can see that this relationship fall into actually three categories. There is a group here in the left side, right side, far right side, there is another group, and you can see a third group in the middle of the curve. What might be the reason? So it points towards, at these three regions of the water, it may be three different processes taking place. So to get a good picture, I looked at the isotopic values, and you can see the N15 values here, which is, if you compare, if you look at these values, they are less at the surface. For example, they are very close to the, they are a little bit lower than the atmospheric values. We know that atmospheric values were 6 point something, but here it's a little bit lower. And oxygen had the same trend as N15 values. So N15, then I looked at the O18 values, as you can see here. O18 values here. All these values showing that there is a nitrification or with a possible second process in the deeper oceans. So the depletions of N2O, N15 and O18 is, at the beginning, at the top of the surface, it's a little bit a slight depletion, but there are some other regions where is very big depletions. But if you have depletions and enrichments in O18, that is due to coexistence of a third process called denitrification, a coupling process, especially in the surface mixed layer. But if you have enrichment in both N15 and O18 at the bottom less oxic layers, which is in relation, also in agreement with the N2O reduction, that is a little bit consumption of N2O. So altogether, this point towards more than one pathway existing in these regions. Then I looked at the isotopomer values. Have a look at this. My isotopomer values at the surface are close to 20. Some of them are they're like, you can see the a little bit increase is there towards the bottom. It's not consistent. It's not the same. It's not like a curve, like straight line from surface to bottom. There is a change in the trend. It can be also due to, it is also due to coexistence of more than one pathway. From all this, I did another experiment, incubation study. What is that? I used labeled isotopes of N15. And what I did, that is for ammonium chloride and ammonium nitrate. Why I used ammonium chloride? If I add ammonium chloride, that will be utilized for nitrification. If I add potassium nitrate, that nitrate will be utilized in the denitrification. So if I have no change in N15 signature for my ammonium chloride after a period of time, that means no nitrification. If no change in N15 value of nitrate, that means there is no denitrification. So there are contrasting results, but finally the results prove that there is no change in N15 for the N15 values of, if you look at here, as I told you, the uptake of N15 ammonium into N2O is an evident from that added from ammonium chloride. So what is that? That means that N2O got N15 from ammonium chloride. That means through nitrification. If I had the opposite, then that is what? Then that is a significant result. I'm getting N15 in my N2O from the potassium nitrate. But in my case, I found a significant increase in N15 values of N2O or when I used to put ammonium chloride. In my incubation chambers where I used potassium nitrate, I did not see any significant increase. So this again proved that there is more nitrification, or the major pathway is nitrification than the denitrification. I just give you a clue of what I have done, and all my results now point towards what? The nitrous oxide. The area of the Arabian Sea is an important source for nitrous oxide to the atmosphere from both the flux values and from the saturation values. And the isotopic signatures from my studies shows that this water's nitrification is the most important pathway than any other process, denitrification or nitrifier denitrification process. So these are the major insights I received from the from my studies. So when you see like how you say this region is very important source, I look at these values. This is the flux of nitrous oxide in teragrams per year, and we have already calculated values of teragrams of nitrous oxide per year from the whole global oceans. So when I compare to that global oceans, this falls into around 20 to 25% of the world world flux. That is a quite large number, and that's why we can say that. What else? So that is my major findings. And since we are talking about the environmental management, I thought to talk about this. This is not my area of study. I did not, especially, the mitigation strategies. So we know now the world oceans are contributing a lot to the nitrous oxide concentration in the atmosphere as compared to the old days. But we know the natural process will not change, as I told you in the beginning. So what might be the reasons? Then only we can think about the mitigation strategies. Maybe Dr. Mishra might be a good person to add on this actually. So reducing the nitrogen fertilizers used in the regions where there is already a large nitrogen surplus will result in a larger than proportional reductions in N2. It's a recent study by Rona. And another thing which I did not mention in my talk is Northern Hemisphere is having more concentration of N2 than Southern Hemisphere. Why? More anthropogenic activities, obviously, more rivers. So more rivers there, the concentrations even into the ocean may be coming. And in the ocean, the coastal regions are all coastal regions concentration will be very high than the open ocean. So and then they also say this is particularly relevant in regions such as East Asia. So the studies really point towards the fertilizers, use of fertilizers, which eventually even reach into the ocean. And there is a very recent another study which also point towards biofuel production might be because it's not much studied, the source as a source of nitrous oxide. So there are a lot of human activities, especially the industrial and the agricultural activities, which contributes to the increased natural sources as well. So we have to think about their reduction. As I told you at the beginning, again, by 2050, a doubling of the concentration is proposed, which is even dangerous than carbon dioxide concentration when we consider on a molecular basis. So see, can we employ the same techniques for other greenhouse gases? Yes. For the other greenhouse gases like methane or carbon dioxide, the major studies are, the concentration studies. Earlier, when I joined this talk, I talk about the studies in the forest. They do flux calculation. They keep gas chambers for continuous measurements. This all we can do after sampling. We can do isotope studies as well for these regions. For carbon dioxide, we will consider the stable isotope C12, and for Oxygen, again, we'll look at the O18. If it is methane, you will look at the again the carbon isotopes and methane isotopes. There are a lot of studies, and this all will give you a good idea about their process, their sources. Because again, the backbone is when a new process is taking place, the isotope values will change in that case. Otherwise, it will remain similar to what the actual source is. So you have isotope from stratosphere, you have carbon from stratosphere to troposphere. Then the tropospheric carbon will have a different signature than the stratospheric carbon if there is an additional process in troposphere or stratosphere. So it depends where you are talking about. Then nowadays, genomics, biology people will be more good at it. Genomic studies are very widely used for, that's the most advanced studies. You study genomic study experiments, you do incubation experiments, you do culture experiments, and you can comment more on the bacterial and biological processes that exist in the corresponding environment. So I would like to, even this work is from my PhD. So I would like to thank my professors, my old university, our excellent Indian research vessel Sagar Sambada and its captains. Also, I would like to thank my people. Plus, I would love to thank you, the team EnACT, for inviting me here. It was a very quick call, and I was very happy to be a part of this. If you have any questions, I will be more happy to answer. If you have any questions, you can ask me here as well in future. That's all. Thanks a lot.
Good evening, sir.
Yes.
There are a few questions you would like to take.
Sure.
So the first one is, what are the primary reasons for increase in greenhouse gases?
You mean like there are anthropogenic processes, as I told you, there
Correct.
are human activities out there. It depends upon what greenhouse gas you are talking about. So depending upon that will change.
Okay.
So for example, when we focus on my study, just one moment. We'll go to my previous slide. So here are you can see very clear here, these are the sources. So anything that will add production from these sources will be the reason for the increase. So it depends which one. So in case of nitrous oxide, if you do more agricultural activities and do more fertilizing in the soil, that will eventually cause an increase, right? But normally in naturals, when we talk about natural sources, they will not change until you do something else that trigger the process in the natural environment.
Yeah. Thank you, sir. The second question is how to reduce the N2O release by anthropogenic activities.
Yeah, that's my answer. We have to, we have to think of mitigation strategies, any kind of mitigation strategies that will increase the concentration. I told you earlier, nitrous oxide, I told you another thing. Nitrous oxide is a precursor of the ozone reduction. Because when it reaches into stratosphere, it will undergo a chemical process. It will convert it to NO, then it will cause a reduction of ozone. After Montreal Protocol, we reduced the CFCs very much in the stratosphere. So now N2O become more relevant in that process, in that case. So why CFC is reduced? We sit together, we talk about IPCC and a panel sit together, they talk about the different processes and different reasons for this. All the nations agreed on this, some nations didn't agree. But anyway, after this agreement, and they looking at the data and the values from the stratosphere, now they shows that the values decreased. Similarly, we have to think about decreasing. So that's why I show you a slide at the end because I expect this question, because though it's not a part of my my research, actually the mitigation, I put a few points here. This is from other researchers who work on the mitigation strategies. See here, you can see reductions might be possible if you reduce the nitrogen fertilizer use. One example. Another one you can see here, which is by the biofuel production, which is not studied. So we need to study about the biofuel production and their role in N2O production. If it is if it can be listed as another important source, then obviously we have to think about a change in the way we produce the biofuel. Now we are not worried about this. I have seen in almost everywhere where I go, biofuel research kind of things, a very important research. But I don't think, like, even here we have, even in India we have biofuel research, not much in the other perspective. Hopefully in the coming years, maybe because it's already just recently started. Just recently, I mean last 20 years, maybe.
So the next question is there any regulations to be followed in using radio-labeled isotopes in the study?
No, I don't use any. I told you there are two kinds of isotopes, radio isotopes and stable isotopes. Stable isotopes, you just like drinking water, no issues. Okay, radioisotopes, it's very different. It's like a lab very specialized for corona study culture. I'm very scared to go into a lab where they're doing corona culture in any part of the world. Same way I'm scared to go to a lab where they do a radioisotope study, because there are a lot of other things we should be careful. Already we are chemists. Most of us are chemists. We know even working in a lab, having a lot of solvents reduce our average lifetime by 5 years if you work for 50 years. So
correct.
Yeah, so it's very
so there is one very curious question by the participants, like is there any chances that we can reverse the global warming in coming future?
See, what we see here is we are thinking, we are talking, we are discussing, but I don't see any decrease. I have seen even people simply talking about, as deniers of climate change. But you look at the data from Mauna Loa Observatory from, Hawaii. How's the concentration of carbon dioxide? I cannot say they are giving a wrong data to us. I really believe on their data. I have visited that station. They are doing 24 hours monitoring for last many decades. The concentration of carbon dioxide is more than 400 now. See, look at the N2O data. In 2011, when I started my research, the ambient air concentration or the air concentration of N2O was, 313. It is 2011. When I finish my PhD and when I'm about to submit my thesis in 15, it is 320. Now, when I give you talk today, if you search for N2O concentration in the air, it is 331.1 ppb. So these all are increasing. And I'm here in Doha. Earlier, I was in New Zealand. I'm from India. Anywhere, any part of the world, we are observing an increase. We are really, actually, we are experiencing this increase, isn't it? Do you think there is no change in climate? It's a very common phenomenon. I don't think so. There is a huge increase in the temperature. This is obviously due to the greenhouse gases. I don't know. It depends on, like we are talking to the people about social distancing and people going, all part of the world, people don't care. It's not only in India or in any states of India, all over the world, people don't care. The same thing happening, I think, in case of greenhouse gases. We all know it's increasing. We have to reduce many things, but we don't care because we need a lot of food. We need to do more farming, agriculture. We need to use fertilizers. So I don't know where it will stop. I'm not very, hopefully, let's say hopefully we will do a reverse, but there are some studies pointing towards, for example, CFC concentration decreased a lot. So it's a, if we work for it, it will work.
Correct.
Yeah.
So the last question is, there are some particular gases among the greenhouse gases. There are particular type of gases which basically is involved in increasing or the temperature, or is basically helping in global warming. So why are those specific gases are only involved in global warming?
I didn't get you. See, I think if I take your question right, I think it depends. For example, you have one molecule of carbon dioxide, okay, and you have one molecule of methane. When you look at this, I will just quickly go to my first slide. Why? See here, carbon dioxide is 56%, nitrous oxide is 6%. Earlier, it was told that nitrous oxide contribute only 0.1 to 2%. Now it is 6%. Anyway, you know, nitrous oxide concentration, I told you already, it's only 330 ppb, while carbon dioxide concentration is around 1,000 times. How much? 400 ppm. But even if N2O concentration is 1,000 times less as compared to carbon dioxide's 56%, N2O is 6%. Why? That's the answer. If you look at the slide left side, if you consider one molecule of carbon dioxide and you have one molecule of N2O, the capacity of one molecule of N2O to absorb heat is 300 times than that of carbon dioxide. Methane has around 200 times, if my memory is correct about methane. So it depends on many factors. That's called the global warming potential, plus their average lifetime, their ambient concentration in the air. Some molecules have concentration in parts per trillion only, even not parts per billion. If you look at chlorofluorocarbons in this figure, look at this. There, if you look at the y-axis in the fourth graph, their concentration is parts per trillion. So that depends on many factors for each molecule to be significant in terms of its global warming capacity.
Okay, thank you so much, sir, for your time and answering our questions.
Thank you so
Over
much.
to you, Ashwati.
Thank you so much, sir, for such an informative talk. I'm sure the students will benefit a lot from this session. So now both the sessions have come to an end here, and it was really enriching for us. Now I would like to call upon Ms. Bindu Rajguru to give the vote of thanks.
A very good evening to all dignitaries and participants present here. It is a great honor and privilege to propose the vote of thanks on the behalf of Environment Analysis and Conservation team of PCSES. I am grateful to our management, Dr. K.M. Vasudevan, Chairperson and CEO of Mahatma Education Society, Dr. Daphne Pillai, Joint Secretary and Rector, Dr. Priyam Pillai, CEO, Pranav Sir, and Professor A.N. Kuti, our academic advisor, for giving us the opportunity to hold this international webinar. I am very grateful to Dr. Gajanan Vade, Principal, and Deepika Ma'am, Vice Principal of Pillai College of Arts, Commerce and Science for their continuous support and guidance, which have greatly helped us toward the successful organization of this event. I sincerely thank our speaker, Dr. Seema Mishra, for kindly accepting our invitation and enlightening us on importance of effect of climate change on mangrove ecosystem and the importance of rhizosphere organisms. I am also grateful to Dr. Naeem Mullungal for gracing the webinar with his presence and sharing his knowledge on role of nitrous oxide in the global climate change, with reference to marine ecosystem. I am sure both the expert talk have enlightened the participants with the aftermaths of climate change and the importance of taking care of our environment and our Mother Earth. Thank you, ma'am and sir. It is my great pleasure to express a deep appreciation for the support and encouragement received from the EnACT members in the organization of this event. I thank all the dignitaries and participants who are from diverse fields in India and abroad for their enthusiastic and active participation in the webinar and making the event a successful one. I also thank Mr. Nishant and his team for the uninterrupted streaming of this event. Thanking everyone once again. Over to you, Ashwati.
Thank you very much, ma'am. Hello participants. The link for the feedback form will be sent to your registered email ID. And I would request everyone to fill the feedback form to avail your e-certificate. We expect your active participation and support for all the future events. Once again, thank you very much. Wish you all a very happy weekend. Stay safe, stay healthy. Thank you.